Imagine spending like $2 billion in 10 years building this incredibly complex microscopic key.
Right.
And then you finally go to use it only to find out the lock literally changes shape depending on the weather outside.
Yeah, that is unfortunately the incredibly frustrating reality of modern drug discovery.
It really is. And I mean, you can't just exhaustively test every possible genetic change, right? Like every perturbation across every single tissue type. There just aren't enough resources or honestly even enough cells in the world to do that physically.
No, definitely not. It is the ultimate bottleneck. Candidate drugs get tested across this massive range of cellular contexts, organoids, animal models, all before they ever reach human trials.
Right, the brute force approach.
Exactly.
Right.
And yet, the vast majority of these compounds still fail late in development. We simply cannot map the sheer combinatorial explosion of human biology through physical experiments alone.
Which perfectly brings us to the mission of our deep dive today. We are unpacking this absolutely fascinating May 2026 paper from Nature Biotechnology.
Yeah, the TxPert paper.
Right, TxPert, which stands for Transcriptomic Perturbation. And it's this computational model, a latent transfer-based deep learning method designed to predict how a cell's gene expression will change after you make a genetic tweak.
And crucially, it's built to predict conditions it has absolutely never seen before.
Which the machine learning folks call out-of-distribution or ODE settings.
Exactly. Because bypassing that physical bottleneck means we have to simulate cells in silico. But, to understand TxPert, we really have to look closely at the messy biological reality it's trying to model.
Yeah, because historically, deep learning models in this space haven't just underperformed, right?
Oh, it's worse than that. Sometimes highly sophisticated AI architectures get beaten by incredibly simple, completely untrained baseline models.
Okay, wait, I need to stop you there. Because when I read that in the source material, it just seemed wild to me.
It is wild.
You have these massive AI models like Gears or SLAM-BDA trying to predict cellular changes, and you're saying a simple mathematical average was beating them.
Yeah, the mean baseline.
Like, if I test 100 random drugs, the average of all those reactions is somehow a great guess for drug number 101. I mean, if I take an aspirin, I don't get the average effect of chemotherapy and antibiotics.
Well, the human-level analogy breaks down at the cellular level because of this exact mean baseline problem. Consider what actually happens when you perturb essential genes.
Okay.
Whether researchers use CRISPR, which is CRISPR interference, basically using a deactivated Cas9 protein to block a gene and turn its volume knob down.
Right, turning it down.
Or they use CRISPR to recruit activation factors and turn the volume up. The cell panics. You are fundamentally disrupting its core machinery.
So it basically goes into survival mode.
Highly conserved survival mode, yes. If you track the gene expression during this systemic shock, specific genes universally spike.
Like which ones?
Genes related to the vacuole, the autophagosome membrane, the lysosome, they all go up.
Oh, because those are like the cell's waste management and recycling centers?
Exactly. Meanwhile, functions related to active cell division and metabolism just shut down. The cell shifts from growth to a state of quiescence.
Wow.
So those previous AI models weren't actually learning the specific nuanced effects of a unique drug. They were just getting highly accurate at predicting, hey, this cell is stressed out.
That makes so much sense. It's like building an AI to predict what song is playing in a busy restaurant, but the AI just learns to predict the sound of clinking silverware because it's the loudest thing in the room.
That's a great analogy.
And if the cell is always screaming and panting, how do you know what actually caused the panic? Especially when the laboratory environment itself is adding even more noise.
Oh, experimental batch effects are one of the most stubborn variables in all of biology.
Yeah.
Biological systems are incredibly sensitive to their environment.
Right.
Slight temperature change in the incubator, differences in how a technician handles the pipettes, microscopic variations in the reagents. I mean, these all alter gene expression.
Yeah, the paper analyzed the Perturb-seq dataset, and the numbers on this were staggering. The correlation between unperturbed control cells across different experimental batches was significantly lower than the correlation between perturbed cells within the same batch.
You really have to let that sink in. The day the experiment was run had a bigger impact on the cell's genetic readout than the actual genetic intervention being tested.
That's just mind-blowing.
Because perturbations are applied to pooled groups of cells in these massive screens, the specific batch and the perturbation ID become inextricably confounded.
They get tangled up.
Right. If an older model used a global average for its control baseline, it was likely mistaking the background noise of that specific Tuesday afternoon in the lab for an actual drug effect.
So how does TxPert bypass this? It strictly relies on batch-matched controls, right? It isolates that specific whisper by recording the exact background noise of that specific room on that specific day. But if the old models were essentially just predicting generic stress and batch noise, what is the new yardstick for success here? Like, how do you even penalize an AI for just guessing stress every single time?
Well, the researchers threw out the old metrics and adopted two highly specific ones. The first is Pearson delta.
Okay, Pearson delta.
Instead of just looking at the absolute gene expression, Pearson delta measures the correlation of the log-fold change.
So it mathematically isolates the difference between the perturbed cell and its specific batch-matched control.
Precisely. And the second metric is retrieval, which asks a fundamentally harder question.
Which is?
Can the model look at the predicted outcome and successfully distinguish a true biological replicate from a completely different drug perturbation?
Right. An unfiltered Pearson delta ended up retaining the specific delicate signal of the perturbation much better than just, skimming the top differentially expressed genes.
Much better, yeah.
So, if you're listening to this and thinking about the messy data you deal with in your own work, you've probably faced this exact paradox. You try to spot a meaningful trend, but realize you're just measuring background noise or systemic stress.
It happens all the time.
Just a quick recap for you before we move on. 1, testing drugs causes a generic stress response in cells. 2, lab batch effects often obscure the real data. And 3, Pearson delta is the metric we use to cut through that noise.
Which brings us to the real architectural challenge.
Right, the AI itself.
Once you identify that generic noise, how do you build a computational engine that actively filters it out?
Let's look under the hood of TxPert. Think about translating a highly technical book. To do it accurately, you need 2 distinct tools.
Okay.
First, you need to understand the native language the book is currently written in. Second, you need a highly nuanced dictionary to translate the specific words. And TxPert uses a process called latent transfer to combine these 2 steps, using 2 separate modules.
Exactly. And the first module is the basal state encoder. This captures that native language, the unperturbed gene expression of the batch-matched control cell.
So it takes a snapshot of the cell before anything happens to it.
Right. It takes that massive, high-dimensional biological data and compresses it into a low-dimensional latent embedding.
And they used an MLP for that, right?
Yes. After extensive hyperparameter tuning, they found that a multi-layer perceptron, or MLP, worked best here. It strips away the noise and provides a clean, mathematical representation of the cell's baseline state before any drug even touches it.
And then the second module is the perturbation encoder. This is the dictionary. But instead of relying on one single source of truth, TxPert uses multiple knowledge graphs, or KGs, of gene-to-gene relationships.
Powered by graph neural networks.
Right. Here's where it gets really interesting.
Relying on a single biological map is just dangerous, so they layered 4 distinct ones. The first is STRING.
STRING.
Yes. This is a massive, curated database of known biological pathways and literature. Think of it as the established biological textbook.
Okay, the textbook.
The second is the Gene Ontology, or GO graph.
And the paper mentions the GO graph uses the Jacquard index to link genes.
It does.
Which, in simple terms, the Jacquard index is basically an equation for overlap. It looks at 2 different genes and asks, hey, how much of your biological job descriptions actually match? It's like comparing 2 resumes to see if they belong in the same department.
That is a perfect way to frame it. So that provides a functional map. But the textbook and the job descriptions are both human curated, right?
Right. They have human bias.
So the last 2 graphs are derived from raw, large-scale empirical screens. PxMap comes from phenomics, which is high-throughput microscopy on human endothelial cells called HUVECs.
And we should probably distinguish that from transcriptomics. Phenomics is visual. It's taking thousands of pictures of cells and measuring their physical morphology.
Yeah, exactly.
Like how stretched out they are, how they cluster together, their structural shape.
Exactly the distinction to make. Phenomics provides structural, visual evidence of cellular changes. And finally, TxMap provides the transcriptomic data.
The genetic wiretap.
Right, counting the actual mRNA molecules inside the cell.
But, I mean, biological data is notoriously contradictory. Some roads on those maps are just dead ends. Some connections in the string database might be completely outdated.
Oh, absolutely.
So how does the AI read 4 overlapping, messy maps without just getting totally confused?
That requires specialized graph architectures, primarily GATT, which stands for Graph Attention Networks.
I always visualize GATT as like a highly intelligent noise-canceling microphone.
Like that.
Because if a central gene is connected to 50 other genes on the map, GATT dynamically weighs the importance of each connection. If it detects a noisy false positive edge, like a bad connection on the map, it actively turns down the volume on that specific pathway and amplifies the reliable ones.
Exactly. It filters the noise, but it still struggles with missing connections.
Yeah.
If two genes interact in reality, but humans haven't drawn the road connecting them on the map, standard networks fail.
Oh, I see.
That's why they introduce XFormer, a type of graph transformer. XFormer uses virtual nodes to allow distant parts of the graph to communicate.
So it can leap over the gaps.
Yes. It can see the hidden highway connecting two distant biological cities in a single computational hop, even if the local roads aren't drawn in.
And to fuse all these maps together, they use multigraph variants like XFormer MG or Gaytime LG, multi-layered graph networks.
Right.
It essentially layers the textbook, the functional index, the visual microscopy, and the genetic wiretap all on top of each other, letting the AI cross-reference them in real time.
It's a very robust system.
So, recapping again for you listening, we have the basal state encoder grabbing the before picture, the perturbation encoder acting as the dictionary using 4 different maps, and attention networks filtering out the bad data. So if you had to predict a complex outcome in your own life, would you trust 1 perfect textbook or 4 slightly messy, overlapping sources of real-world data?
I'd take the 4 messy sources every time.
Right. So we have the baseline cell and we have our multi-layered dictionary. Let's look at the ultimate test of intelligence here, which is predicting data it has absolutely never seen before.
Because generalization is the true bottleneck of AI.
Yes.
The researchers put TxPert through 3 massive out-of-distribution tasks, comparing it against competitors like Gears and SLAM-BA. Task 1 was predicting unseen single perturbations across 4 heavily studied cell lines.
That's K562, RPE1, HepG2, and JERCAT, right?
Yes.
And assuming you know your cell lines, the performance here was solid across the board. TxPert uniformly beat the competitors and the generic mean baseline. But the benchmark they used to measure this completely blew my mind.
The split-half experimental reproducibility.
Yes. Explain that, because it's wild.
It goes back to the intrinsic messiness of biology we were talking about. If a human scientist does the exact same experiment twice, under the exact same conditions, the resulting gene expression profiles will not be identical.
Right, they never are.
The Pearson correlation is far below 1.0. So to find the absolute ceiling of possible algorithmic performance, the researchers literally took the real human lab, replicate data for a specific perturbation, and split it down the middle.
They tested one half of reality against the other half of reality.
Exactly.
And in K562, JERCAT, and HepG2 lines, TxPert's predictions were astonishingly competitive with that split-half benchmark. It's essentially matching the consistency limit of biological reality itself.
It really is. But Task 2 pushed it further into this combinatorial nightmare, double unseen perturbations using the Norman dataset.
Okay.
What happens if you perturb 2 genes at exactly the same time?
Which is incredibly complex, and the standard fallback for older models is an additive baseline. The logic there assumes that if perturbing gene A causes effect X, and perturbing gene B causes effect Y, then doing both just equals X plus Y.
But why wouldn't biology just be additive?
Because it's synergistic, right? Genetic pathways constantly interfere with and amplify each other.
Exactly. 2 drugs might target the same downstream protein, canceling each other out, or they might trigger a feedback loop that causes an exponentially larger reaction.
And TxPert caught that.
Yes, TxPert, specifically using the JAG-LD architecture, substantially beat both the additive baseline and the Gears model. It successfully mapped those synergistic interactions.
Which leads us to task 3, cross-cell line prediction. This is the hardest test by far. You ask the model to predict a perturbation effect in a cell line where 0 perturbations of any kind were included in the training data.
And this is where latent transfer proves its worth. TxPert exceeded the non-learned general baseline in all 4 held-out cell lines, consistently beats SLAM-BDA, and even beat a highly challenging nearest cell line baseline in K562 and RPE1.
But wait, mathematically, how does it translate rules from, say, a leukemia cell to an endothelial cell if it's never even seen the latter perturbed.
Because the architecture cleanly separated the basal state encoder from the perturbation encoder.
Oh, the native language versus the dictionary.
Exactly. It learned the verb, the mechanism of the genetic tweak, independent of the noun, the baseline cell. When confronted with a new cell line, it maps the new baseline language and seamlessly applies the biological verbs it learned from the K562 cells.
That separation of variables is brilliant.
It's very elegant.
So summarizing this part for you, TxPert crushes single unseen predictions, handles double gene combos better than simple math, and generalizes to new cell types. We often talk about AI replacing human tasks, but here, the ultimate benchmark is split-half reproducibility, basically the ceiling of how consistent reality is. How do you measure success when the ground truth itself is messy?
Well, to really trust in AI, we can't just applaud its wins. We have to try and break its maps.
Right, ablations. The researchers performed rigorous ablation studies specifically for this reason. They took the string graph for the K562 dataset and deliberately started rewiring the edges.
They forced connections between genes that have nothing to do with each other, corrupting the map from 0% all the way up to 100%.
And as the map got worse, the model's test set Pearson delta steadily collapsed. Taking away edges, downsampling the graph, also crippled performance.
Which tells us something important.
Yeah, the conclusion is undeniable. Accurate biological graphs are the absolute secret sauce of TxPert. Incrementally adding all 4 graphs together yielded the highest peak performance.
But relying on human-curated maps introduces a really dangerous bias. The researchers evaluated the model using the Pharos knowledge rank,
Right.
which categorizes genes into bins from 0 to 5, based strictly on how heavily researched they are in the scientific community.
And unsurprisingly, TxPert predicts outcomes significantly better for genes that are incredibly well-researched. But wait, if it only predicts well on things we already know about, is it just regurgitating popular science?
That's a very valid critique.
But how is it discovering anything novel?
The curated graphs, like string, inherently carry the bias of human attention. But the architecture provided a failsafe.
The phenomics data.
Exactly. The researchers found that adding the empirical PxMap graph, the visual phenomics data generated by machines, not human literature, boosted the model's performance across all knowledge levels.
So the raw experimental data acted as a patch for the holes in our human bias.
It did. But even with that patch, we have to highlight a massive, glaring failure mode.
Oh, right. The paper gives the specific example of the DYNC1H1 gene.
Yes. This reveals a fascinating algorithmic blind spot. When you perturb DYNC1H1 with CRISPR interference, TxPert is highly accurate at predicting the complex downstream ripple effects throughout the cell. It accurately maps how protein translation and distant pathways shift. But ironically, it completely fails to predict the standard downregulation of the actual target gene itself.
That is crazy. It perfectly predicts the ripples in the pond, but completely misses the splash of the rock. Mechanically, why does it miss the most direct, obvious consequence of the intervention?
It's a known flaw in graph neural networks called oversmoothing.
Oversmoothing.
Yeah. The AI relies heavily on a gene's neighbors to determine its state. If all the surrounding genes in the network are screaming stress, the model assumes the center node must also be highly active.
So it smooths the data.
Exactly. It fails to realize that the center node is silent precisely because the CRISPR intervention forcibly turned it off. The AI over-relies on the network consensus and ignores the direct biological intervention.
It's so counterintuitive that many researchers actually just hard-code the target gene's value into the final output because the AI fundamentally struggles to predict its own primary target.
Yeah, they just have to patch it manually.
Yeah. It's a great thought experiment for anyone managing complex systems, you know?
Yeah.
Do you have blind spots in your own expertise where you perfectly anticipate the complex downstream effects, but somehow miss the direct impact of the initial action?
Yeah.
So just wrapping up this section for you. Messing up the graph data ruins the AI. Combining all 4 maps is best. The model favors well-researched genes, and it totally whiffs on predicting the drop in the specific target gene.
It's a humbling reminder that these models, as advanced as they are just mathematical approximations of reality. They carry the biases of their algorithms and the limitations of their maps.
So let's bring it all together. We started this journey looking at the impossibly expensive physical bottleneck of modern drug discovery. We explored how TxPert attempts to bypass this physical limit by fusing diverse biological maps, textbooks, functional indexes, microscopy, and transcriptomics.
And by using latent transfer to separate the cell's native state from the mechanism of the drug.
Right, and using attention networks to filter out background stress and batch noise, it can accurately simulate cellular responses to entirely new genetic interventions.
The implications for the next decade are just massive.
Yeah.
Right now, these benchmarks were mostly tested on immortalized cancer cell lines, which are relatively homogeneous.
Right, the easy stuff.
But as the field moves toward modeling primary human tissues and begins to integrate spatial transcriptomics, like how cells interact with their physical neighbors in an organ, these in silico virtual assays will fundamentally alter the economics of screening.
We are looking at a critical stepping stone toward true personalized medicine.
Absolutely. A future where we can test 1 million therapeutic combinations on a digital twin of your specific cellular architecture before you ever take a single pill.
It is an incredible leap forward, but it leaves me with a provocative final thought for you to mull over. TxPert's predictive power relies entirely on the maps of biology we currently possess. But if these AI models become the unquestioned gold standard for discovering new drugs, and their predictions are strictly constrained by the graphs we feed them, how do we ensure we don't trap ourselves in a feedback loop of our own existing biological biases? If the map makes the medicine, who is making sure the map is right?
Well, a powerful engine will just drive you into a wall faster if you give it the wrong coordinates.
Exactly. We all want that clean X-ray, that perfect biological lock and key. But maybe the future's medicine isn't about finding a single clean line. Maybe it's about learning how to translate the noise.